Lucy is the first who is starting the presentation, but just if we can settle the slides.
We're here a high-tech bunch with type 1 diabetes. I'm sure we can figure out the screen sharing. Hello, everyone, and thank you so much for your time today. Really appreciate you making the time. So just do let me know if you can see the screen and if everything, yes, perfect, great. So my name is Lucy, and thank you so much for the introduction. I'm part of the Levicure team, and we are a startup in Israel that was founded by Dr. Shmuel Levit. It is important to say that first and foremost, Doctor is a clinician with 35 years of experience in treating people with endocrinological disorders. And within his own private medical practice, he was able to actually establish that a combination of two very well-known and approved FDA drugs and a supplement GABA can lead his type 1 diabetic patients to significantly reduce their insulin demands, and in some cases, wean them off insulin completely. So if you're familiar with academic research related to drug development, you'll probably remember that it usually goes from cells to animal models, and then if you're really, really lucky, to humans. So our case is quite special precisely because Dr. Levit was able to see the effectiveness of the triple combination of GABA together with DPP-4 inhibitor and PPI on 19 of his adult patients and within his private clinical practice. So he will go over the detailed findings, but I can say that the most promising observations were in the early onset group, meaning that they were diagnosed less than 12 months before the treatment began, and with 7 out of 10 patients discontinuing insulin therapy completely. And there was also a significant increase in detectable c-peptide. So I'm happy to share that this study undertaken together with Uppsala University was just accepted for publication in one of the leading peer-reviewed journals, and will be published in the coming few days. And I'm sure our friends in T1Cure will help to circulate it to anyone interested. So in type 1 diabetes research community, there is a very popular saying, and it goes that we know how to treat diabetes in mice, but it never actually translates into type 1 diabetes in humans. So here I think we're at a great advantage because the study gives us confidence that these findings are going to translate very well into human phase 2 trials, simply because it's not the mouse model that we're looking at here. The triple therapy has been successful in humans with type 1 diabetes. So not as exciting, but we also did a proof of concept animal model study for treatment and prevention on NOD mice from Jackson Labs, comparing the triple combination with all the double therapies, including those previously researched and placebo. And this was primarily needed to secure patents in the US and Europe. And I'm also very happy to say that these results corresponded really well with Dr. Levit's findings on humans. So you see how we go back round. And the publication is already out in Frontiers Endocrinology and is available to anyone interested. So based on our observation and analysis of the existing research with incredible input from our scientific advisory board and Uppsala University, we're able to form a hypothesis on the mechanism of action. Unfortunately, I don't have time to fully cover it in detail, but we'll try to give an overview of our vision. And I think it's also a really good time to briefly introduce the team, which is really the heart and soul of Levicure. Next slide, please. So I begin with Dr. Levit, our founder and the man behind therapy. We're also incredibly fortunate to have Dr. Alexander Rabinovich, a professor of medicine and former director of diabetes research at the University of Alberta. He dedicated his entire career to diabetes research, specifically focusing on combination treatments, including DPP-4 inhibitors and PPIs, and of course, Dr. Helge Schiot, who is a teaching professor and head of pharmacology and neuroscience division in Uppsala University. Dr. Torban is our experienced clinical endocrinologist with 15 years of experience as an investigator for clinical trials for biggest pharma companies in the industry. Now, Dr. Yeshit Stark is a former VP and head of global clinical development at Teva, with 34 years dedicated to clinical research and development, and the very woman responsible for Teva's flagship drug, Copaxone, intended for multiple sclerosis. Mike Tyler is also former Teva VP on generic and R&D with 35 years of pharma experience and extensive expertise in drug development. The former executive Teva team would not be complete without our strategic and executive advisor, former VP of Teva Europe, Amas Anasot, and our management and operation is in capable hands of Daniel, who's our CEO with 12 years of executive experience in biotech startup, and Valentine, former director at private equity fund with over 10 years of project management and experience. So my function is primarily involved in scientific research, legal counsel, and assisting our advisory panel, while Olga runs all day-to-day operations. So thank you, and next slide, because by now I'm sure you wonder how and why the treatment actually works in type 1 diabetics. So let's get into it. And as you all know, type 1 diabetes, and as has been kindly presented by Ben, is an autoimmune and endocrine disorder. And the classic understanding of the disease is that autocorrective immune cells mistakenly attack and destroy the healthy beta cells. We propose an alternative view of type 1 diabetes where there are several dysfunctions that happen simultaneously in immune and endocrine systems, and both need to be effectively addressed for any potential treatment really to be effective in managing the disease and hopefully achieving remission. So on this slide, if you look really closely, you'll see two people. One is healthy, if such thing even exists, and another a type 1 diabetic. So I will begin by saying that immune and endocrine cells are very similar to neurons in how they're regulated by and can release neurotransmitters. And we are interested in two. One is glutamate, which excites, and the second one is GABA, which inhibits. So they are driving forces for the immune cell behavior. And in endocrine cell, their balance actually regulates the beta cell survival and insulin secretion. So beta cells of the pancreas, again, as I'm sure you know, are especially fragile with limited means of self-defense should something go wrong. And to simplify, if there is too much glutamate and breakdown in the GABA transmission, this creates a really toxic environment that leads to extreme stress and eventually a beta cell death, even without the involvement or assault from the immune system. So let's get back to the image, and we begin with looking at these two people with their immune cells in the resting state, and before type 1D diagnosis is apparent. So our hypothesis is that in type 1, there will be a pathogenic process already in motion, setting the toxic environment, such as elevation of glutamate, serum DPP4, ADH, to name just a few. But they are not evident because the immune cells are sleeping, if you will. So then a trigger event happens that activates the immune system, for example, a virus. And many type 1D patients will report that they were diagnosed after such trigger event. And immune cells on this slide are activated in both individuals. However, in type 1 diabetic, there will be a toxic environment as a driving force to which the cells wake up to, and the elevated markers begin driving and altering their normal behavior, making them proliferate more, release more cytokines, and also prolong their survival. So now we have activated the autocorrective immune cells that are attacking the pancreas that is already suffering from excitotoxic stress, which leads to gradual beta cell destruction on both fronts. So how will our treatment work in these conditions? So next slide, please. Well, as you probably know, the current approach of big pharma is finding a treatment that is focused on looking for that single magic bullet that is going to solve everything and for everyone. And it's probably not going to be effective if you have at least two moving targets. So type 1D is a very complex disease that has several different dysfunctions that need to be addressed, like autoimmunity, elevation of glutamate, to repeat myself, elevation of DPP4 being just a few. And they are different in the way that they affect the cells and manifest eventually into type 1 diabetes. So therefore, a brand new molecule to patent that is, of course, very lucrative, and that can be a cure for everything, apparently. As you can see from this illustration with lots of arrows, would have to target a lot of different processes all at once. And that's extremely hard to achieve. So we believe another way would be to build on the existing understanding and knowledge of the existing drugs that are well known and that can target each of these pathways individually and work synergistically together, such as DPP4 inhibitors, PPIs, more specifically sitagliptin in our case and omeprazole, that had been extensively researched for decades and are recognized as safe and readily available drugs. And we believe that the combination of these two components together with GABA, that has also been incredibly widely researched for type 1 diabetes, and now is actually a very hot time for GABA. So they can work together synergistically and target all these different pathological processes that occur during type 1 diabetes and have a significantly more efficacy compared to the double combinations or single components alone. So I will be happy to answer any questions on the mechanism of action in detail separately, but for now, I think I should, and I'm very pleased to give word to Dr. Levick to cover his clinical findings and describe to you exactly how therapy works in real patients. Thank you.
Good day, everybody. My name is Dr. Professor Levitt, and I just have to say a few words regarding my past. My great teacher was my father, who was the prominent scientist of the former Soviet Union, and he was immunologist and endocrinologist at the same time. So 40 years ago, we studied the autoimmune diseases, and we studied them from the point of the possibility of reversibility of autoimmune endocrine diseases. And my doctorate is devoted to the reversibility of Hashimoto thyroiditis, which is classical model of the autoimmune endocrine diseases. And 40 years ago, I knew that we treat the autoimmune diseases, we can reduce or even reverse the autoimmunity and try and succeed preventing our autoimmune disease in our patients. So it took several decades until I decided to try it in type 1D patients. And first of all, I changed the understanding and the concept of type 1 diabetes, which is now my attitude to this disease is the kind of neurodegenerative disease, as Lucy previously said, because the processes are very similar to the processes pathogenesis in the brain diseases. So we studied 19 patients in our clinic who actually get the triple therapy, and we also divided them to the early patients, which means they get their diabetes within 12 months from the beginning, and late therapy, where we are talking about 9 patients who actually suffered from diabetes for a long time. This is the prolonged and advanced disease. So we can see that the differences were absent within the group, beside the duration of insulin therapy, which was remarkably much longer in the late therapy group, and it is logical. Now, next slide, please. The main term of protocol was the duration of the therapy for 26 weeks at least. So these are main results we get. I have to be as short and brief as I can. So this is the summarizing slide, where you can see the result we get. The first graph on the left side shows us the fasting blood glucose, and we see also the early therapy group. I just want to remind it is 12 months of the beginning, less than 12 months from the beginning of diabetes, and late therapy or advanced type 1 diabetes, more than one year after the beginning. So we have got the significant reduction of fasting blood glucose in both groups. Regarding A1C, the measurement of the diabetes control, we received the very impressive reduction of A1C in the early therapy group, but we didn't get the reduction of A1C in the advanced diabetes, and I'm talking about the absolute numbers. But when we used the specific metric named insulin dose adjusted A1C, as you see at the picture C, we also get the reduction of the A1C as well in the late onset group, which is remarkable because according to the classical attitude, there is no beta cells and there is no substrate to influence and to affect by the therapy. And nevertheless, we get the results, which were impressive. And first of all, it was expressed by the significant reduction of the insulin dose. it is worthwhile to emphasize that even in the late onset group, it was the 38 reduction of insulin demands, which is incredible, and 69% insulin demands was noticed in the early onset group. And if we talk about daily insulin dose, as it is reflected on the graph D, we see the reduction was highly significant in both groups. So regarding the insulin weight ratio, which is very, very important parameter and metric, we also received the very significant reduction in insulin weight ratio in both groups, in early group and in late or advanced therapy group. And the main confirmation of the successfulness of our attitude and or our approach is the elevation of c-peptide. It is worthwhile to emphasize here that the elevation of c-peptide was very significant, 2.5 times more than the basal one in the early therapy group, while there was no c-peptide elevation in late therapy group. So we explain the clinical improvement in the late therapy group in spite of the elevation of c-peptide by other mechanisms which are actually involved in the regulation of insulin sensitivity and insulin demands in these advanced patients. And we still consider the late onset of type 1 diabetes may benefit with this triple therapy. To conclude and to summarize, we've got the in early onset therapy group, we've got the reduction of fasting blood glucose by 25% and reduction by A1C by 38%, reduction in insulin demands by 69%, which is incredible. But the main success was the 70% of insulin winning in our late onset group, because these are actually incredible results. This is at the level of unbelievable, because this is of course not a so-called honeymoon, because honeymoon is a very short period and it is very rare where our patients discontinue the insulin. In spite of the honeymoon, it was very prolonged discontinuation of insulin, and we still have some patients that for years without the insulin continue to live without insulin, receiving our triple therapy. Now, the increase of c-peptide, as I previously said, in the early therapy group was 145%. And regarding the advanced type 1 diabetes, it was still very impressive results, expressing by the fasting blood glucose reduction by 19%, and reduction of the insulin demands by 38%. All in all, it is a confirmation of the effectiveness and safety of our triple therapy approach. Thank you very much for your attention. And if you have any, just one moment, I have to emphasize the successful patients here, and to notice the 6 patients actually discontinued the insulin, while 2 of them are still without insulin, and the average duration of remission without insulin was 17 months, which is very, very impressive. Next slide, please. Okay, in order to demonstrate the successfulness of our treatment, the CGM is the best way. And here at the left side, we can see the patient before starting our treatment, who actually was treated with 12 units of insulin. His A1C, basal A1C was 8.5, and fasting blood glucose 130. And after 29 weeks of triple therapy, we see here on the right side the curve, which is compatible with a healthy person. you should not be an endocrinologist or a doctor to understand, in order to understand that this is the huge difference between two graphs on the left and on the right. And this is the completely healthy person with 5.2 A1C, fasting blood glucose 100, and 0 units of insulin demands while treated by triple therapy. Next slide, please. Another patient who is 12.1 A1C at the beginning and c-peptide 140, and after 27, I was made to prescribe him the insulin because he was a glucotoxic patient. He was very high blood sugar. And after several weeks of triple therapy, we stopped his insulin. And on the right panel, we can see the 27 weeks of triple therapy, 0 units of insulin demands. C-peptide rose from 140 to 210, and his A1C was reduced from 12.1 to 6.2. Next slide, please. Okay, so let us summarize. We're talking about repurposing of the FDA-approved, well-known, safe drugs, which are in triple therapy and combined in this specific combination have a very, very impressive effect on the type 1 diabetes, and in our opinion may reverse this disease and suspend the long-term remission. Sometimes we are talking about years of remission. Thank you for your attention.
Thank you, Dr. Levitt. I will now continue maybe with not that interesting part, but also very important part for actually our drugs. As Lucy already told you that humans were the starting point, but to obtain our patents and to make this company possible, result of our NOD mice experiments was also of vital importance. What we obtained, we looked at 5 groups, we looked at triple therapy against all double combinations and the placebo. It is important because only the triple combination worked in the severely type 1 diabetic mice, which is NOD mice and the gold standard for the industry. We also have seen that the c-peptide levels rose significantly tenfold only in a triple combination group. And the same we have seen for the insulin in blood plasma. Why it is critical? Because we have shown that it is only the synergistics of the 3 targeting all the pathogenous basis of the disease, we are able to achieve significant and sustained effect on treating of type 1 diabetes. Based on this study, which we actually published in the Frontiers, and Ben probably will distribute it after the call, we were able to obtain our patents. We have been already granted the patent in the United States and Europe for the treatment of type 1 diabetes with our triple combination therapy. We are not standing on that way. We are also extending our pipeline for our future investigations. We also applied for a provisional patent in the United States for quadruple combination. And we are also running the quadruple combination experiment, which shows even more impressive results and could be a drug for an advanced patient treatment. Based on the mechanism of action, which was developed by our team, we were able to apply for a patent in neurology to target with the same drugs other autoimmune and neurological disorders with similar pathogenic processes. And now I'm coming to our strategy and our main goal. Our main goal is to launch the triple therapy by 2029 and bring type 1 diabetic patients worldwide the effects of the therapy which Professor Dr. Levitt just described to you. And a bit of our history. So we were founded in 2020 with a seed money investment to complete the mice study. We also did the analysis of Dr. Levitt patients. We identified the ideal regulatory pathway, and we applied and were granted with the first patent in the United States. Last year, in September, we were able to secure funding from 5 angel investors who are big supporters of T1D community. And the goal of this pre-series A round of funding, which we are finalizing all the activities at the moment, was to confirm and create the clinical and CMC development strategy and confirm it with the FDA, with American body, and with EMA, with the European body. Why it is so important? It is important because once it is confirmed, we can proceed to our Series A round of funding, and we will clearly know how much money we actually need to execute the strategy. In this activities, we are supported by an amazing team of Connexicum consultants, which are founded and headed by Zan Fleming, who is a former FDA expert himself. Also, we were able to strengthen our IP portfolio, and we are publishing our research and works. We have just published NOD mice trial. The human data which Professor Levitt described today was just accepted for publication and probably will take additional 1 or 2 weeks to be able to read it online. Mechanism of action is nearly finalized for publication. Also, as I told you, we're running the 4-component therapy experiment. All these pieces, all these puzzles together, our IP, our patent, the confirmation of our clinical and CMC strategy from FDA will be the foundation and all the results which we show today for our Series A round of funding, which will focus on the phase 2A clinical human trial. And we have now here Mike Taylor, who will describe you all the details of our activities in the phase 2A clinical trial in Series A. Mike?
Hi, Daniil. Can you hear me?
Yes, we hear you well.
Okay. So you want me to talk about the Series A?
Yes, please describe all the activities. Yeah.
All right. So yeah, as Daniil said, we've been very busy planning for the clinical trial, the phase 2A trial, and all of the activities that need to happen in advance of and in order to facilitate that trial. First of all, we need to, as Daniil said, we need to make the submission to FDA and get the pre-IND advice from FDA so that we know that we're on the right track. If we have to make any modifications to the synopsis, to the planned study that we've put together, we'll do that after getting FDA advice. We have to line up a clinical CRO. So we've been talking to 3 or 4 different clinical CROs who can help us to manage the study. These are full service CROs. And when I say full service, of course, full service means different things to different people. And so we will have to add different activities around the full service that the CROs provide. But they will be lining up the different sites. They'll be getting the investigators for us. Those sites will be responsible for recruiting the patients. We'll have, they'll be doing the technical writing for the full protocol, and then of course doing the biostatistics to assess the results. They'll also arrange for a central lab to test, the blood samples, and of course we have to look at the safety. We have to look at other physiological parameters to make sure that our patients also remain safe. But before we can go into the clinical trial, we're going to have the drugs that we need in order for the clinical testing, the investigational products. So we have 3 drugs, as we've already described, and we're going to need 3 placebos because this is a randomized, prospective, and placebo-controlled trial. So we need to create those 3 placebos. Those don't exist. We've been talking to 3 different potential organizations that can all do that clinical supply manufacture for us. And we will have those, we will have all of those products available for the start of the study. One other wrinkle is that while the sitagliptin and the omeprazole are both commercially available drugs, and those are, we can buy those in the market and we can prepare placebo copies of those, GABA is only available as a food supplement. And as a food supplement, while ironically, while it's available freely in any health food store and you can buy and take as much as you want as a dietary supplement, As a drug, it's going to be regulated in a very different way. And it's going to, GABA has never been approved in the United States or in Europe as a pharmaceutical, as a medicinal product. And we know from discussions with our regulatory consultants and with others that both the US and the European regulatory authorities take a much stricter view when you're talking about doing a phase 2 clinical trial than the food supplement arm of the same agency. And so we are going ahead with a food grade, with a supplement grade of GABA, which we're planning to encapsulate in our own contracted cGMP facility so that we will have GMP quality material. But if that's not accepted, then we also have we have 2 fallback plans in order to manage the risk behind that. So if necessary, we can do all of the testing ourselves, and at the same time, if none of that is accepted, we will actually go ahead and produce our own GMP GABA.
Okay, thank you, Mike, for clarifications. We have, I would say, an extensive plan how we will do and the phase 2A trial. I think it is of most important to conduct the prospective randomized trial so that the worldwide the audience and the key opinion leaders and governments and corporations accept our vision, and this would be the cornerstone for the future product. We will also develop a final formulation, which will be 1 product, 1 pill a day. This is our ideal focus for the future, that patients should just receive 1 product, 1 pill a day. And we will also conduct some proof of concept studies for other indications if we can utilize the same product, the same pill for other patients in other diseases.
This truly is a global effort. If you want to support us, please share our information. You can help us out just by sharing us word of mouth on social media, on LinkedIn. Of course, we appreciate any financial support. You can give a tax-deductible gift to T1Cure as a pass-through to Levicure or directly to us to help us continue with our own operations. Again, I want to say thanks so much to everybody for wanting to be part of the solution. So have a great day. Have a great evening. Thank you.
Thank you.
Thank you very much.